Hi, Gray. How are you doing?
I'm great. How are you? And thanks for having me, Adrian.
I'm good. It's a pleasure to have you here. So, Gray, you're a biologist. You're currently an assistant professor at the University of Davis. And so you're interested in understanding the mutational origin of genetic diversity in plants. And so the first question I have for you is, when and why did you decide on becoming a biologist?
So, yeah, that's a good question. I didn't really realize that being like a professional scientist was a career I could do until I started doing some research as an undergraduate student. I was a pre-medical major and kind of had my sights on that career, and then I spent some time in the lab and got to know a professor really well who gave me great mentorship, and he exposed me to the world of research, that you could actually go out and spend a career just asking cool, creative questions. And so it was really as an undergrad, nearing the end of my program, that I kind of came to this realization that this might be something really cool to be able to do, to pursue my curiosity, to ask creative questions and work with really interesting people all the time. So
that's cool. So, and did you get to work on plants directly, or did you just wander around just full of scientific curiosity and just getting to know every kind of stuff, and then there was this plant that got you in love with plants? Or
I mean, I've always loved biology, and the work as an undergrad was kind of plant related. So I had some experience there. Actually, after my undergraduate time, I spent a year working in a mammalian immunology lab, and the research was interesting, but I realized I didn't really enjoy working with mice and rabbits. I don't know if I was cut out for that work. And so I decided, when I was going to go back to grad school, I was going to return to this passion for studying plants. And I mean, plants are amazing. They're just beautiful, feed the world, and they're just cool organisms, and there's lots of interesting questions we can ask with them. So
they're pretty cool. I think what I enjoy most about plants is their green color.
Yeah, they're beautiful.
I just love that.
I mean, we put them in our homes, because they're just nice to see, and they're fun to work with.
So actually, what kind of plants have you been studying? So you started in grad school studying plants. So what kind of plants did you study? Did you study them directly in their ecosystems, or in the lab, or both of them?
A little bit of both. I mean, the main system that we work with is called Arabidopsis thaliana. It's a model system for, it's the fruit fly of plant biology or something like that. It's a model system, little plant that is great for doing genetics, and we can do all kinds of experiments in it. And I'd say that's the main species that we've worked in. And now at UC Davis, agriculture is a big part of the California economy and a big part of plant biology in general. And so we've expanded into looking at different crop species. So, like, for example, we're working on pistachio trees now and thinking about climate adaptation in pistachio. We have work on cassava, which is also known as yucca, important crop for the global south. We've kind of taken some of the things that we've learned in these model systems, like Arabidopsis, and are expanding it to agriculturally important plants as well.
Okay, that's interesting. I guess the kind of time scale of these studies is really variable between a model species and these agricultural species.
Like Arabidopsis can complete its whole life cycle in a couple of months, and a pistachio tree grows for 70 years, and it takes 8 years to produce its first flower. So the way we approach those systems experimentally is, completely different. So
what kind of ways are you taking to approach the systems? Are you genetically characterizing all the pistachio trees, for
something
example,
like that? So we're interested in genetic diversity in pistachio. So there's a breeding program by Pat Brown at UC Davis that we work closely with, who's really putting these things into practice to actually make crosses between pistachio trees to release the next exciting variety for the pistachio industry. And we're kind of on the back end doing genetics and genomics and sequencing lots of genomes and asking questions about patterns of genetic variation within that diversity.
So when you're thinking about genetic diversity, are you thinking of differences in the DNA sequences themselves, or is there also a relationship with other kind of genetic modification like methylation?
Yeah. I think that the interaction between DNA, the variation at the DNA level, and processes at the epigenetic or epigenomic level, when we think about methylation or histone modifications, there's a lot of really cool opportunities to study that, and I guess that's kind of the other big branch of our lab, is thinking about how the epigenome can influence mutation, for example, how it changes which mutations happen across the genome, and how that gives rise to some of the patterns of genetic diversity that we see in different plant species.
So do we have some hints right now about how those two different modifications of the genomes are interconnected, or how they interact together? Yeah,
we have some pretty good ideas about specific mechanisms. So I'll give kind of a visual of it. So we have, if you zoomed in a cell, right, you have DNA, but it's not just randomly organized, it's structured. So you have DNA and it's coiled around these proteins, histones, that form this thing called a nucleosome. And those histones have little tails on them, and that tail can be chemically modified. So it's like imagine putting like a sticker, a red sticker on this histone. And it turns out that those, which sticker it is tells you what part of the genome you're in. And this is really cool because what people have discovered is that those chemical modifications, those stickers that say, this is an important gene in the genome, those things are being recognized by DNA repair proteins that say, hey, I'm an important gene, and DNA repair proteins are recruited specific to that region. And as a consequence, those regions that have that sticker, that mark, they're seeing lower mutation rates because they are recruiting DNA repair preferentially to those regions. And so this has been really well studied in human biology because the implications of these things for cancer emergence is obviously really important. Thinking about cancer as a mutational disease and understanding how that works has driven a lot of the mechanistic work there. And so we've kind of come at it from a plant biology perspective and started asking, well, what's happening in plant genomes? Are they doing the same things? Are they doing something different? And to kind of jump to the punchline, what we've discovered is that plants and animals are doing the same thing, but through convergent mechanisms. So plants have a different sticker, and they have a different reader that recognizes it. So they've achieved the same goal or the same functional outcome, but through independent origins. So it looks like plants evolved a system and animals evolved a separate system. So it's like a really cool example of evolutionary convergence.
That's interesting because it actually means that these kind of systems of DNA repair, the stickers, were actually probably not present in the common ancestral, like eukaryotic ancestral. The complete system, the deposition of the sticker and then the reader probably was not ancestral to all eukaryotes. And it may be that different species and different organisms have evolved different systems. We've studied really one well-characterized model protein system for this, but there's so many unknown unknowns across the tree of life. There's so much interesting diversity that really just hasn't been explored, that we just may not know that they've evolved some other really cool system that just simply isn't known at this point. So do you know? So where do you see your field going in the next 10, 20 years? What are the implications for science or for society of knowing how are we going to be able to predict how any environmental changes like global warming or changes in the light intensity for plants, for example, how that will affect the genomic diversity?
This is a really big question and an exciting one. I think that in terms of advances in the field, there's huge technological advances in DNA sequencing, for example. So our ability to ask questions about mutation at all is really accelerating because we have new technologies that enable us to do that. And then I think in the context of big problems like climate change, there are some interesting sort of ideas and hypotheses that emerge from all of this. So for example, we know that those stickers, the placement of them is dependent on gene expression, is different in different environments. And so it raises all kinds of cool questions about how does the environment shape, these mutational processes. And the implications for climate adaptation are basically unknown. We don't know. It may not be important. It may be interesting. It remains to be seen. But I think the other side of all of this is, of course, in human health and thinking about how DNA repair systems work optimally to prevent disease in different environments or different with different stresses. I think is worth exploring more, which is exciting as well. So.
And it's interesting that you say that how it works optimally. An alternative question would be, is it working optimally?
Right.
And maybe can we use what we learn from plants or other organisms to maybe tune it? I don't know if that could be an outcome.
I think that's a great, is it optimum? Is it already the best it could be? Or has some other organism actually figured out a better system? And we don't see that in this species or but by surveying the diversity of these mechanisms, we might discover some really nifty thing that some group of species has evolved that's really solving a critical problem that doesn't exist in the human genome, for example, but could give clues into useful things that are useful, perhaps.
Perhaps.
It's all very speculative, but fun to think about.
Well, thank you very much, Gray. It was very nice having you here.
Thank you so much.